morning. I'm Xiaoyue. I'm a PhD student at the Duran Lab at Carnegie Mellon. My talk today is on vector semantics of multi-domain protein architectures. Multi-domain proteins are mosaics of sequence segments that encode structural or functional units called domains. These segments are found in otherwise unrelated proteins and can fold into a characteristic shape in many different contexts. For example, this is the MPP6 protein, which contains 5 domains, 2 L27 domains, a PDZ peptide recognition domain, SH3, and a catalytically inactive guanylate kinase, or GLBIO domain. So on the left, you can see that these domains appear as distinct folds in the MPP6 alpha-fold structure. And this is a cartoon representation of the sequence segments that encode the individual domains, and this abstract representation, the sequence of domains in N-to-C terminal order, is referred to as the domain architecture of a multi-domain protein. The domain architecture representation ignores the specific amino acid sequences of the domains and the linker sequences that connect them. It focuses rather on the large-scale organization of protein architecture and modularity. The DA representation has proved extremely useful for studying protein function and evolution. And shown in this box is just a sample of the types of studies that have used this representation. The domain architecture of a multi-domain protein is intimately related to its function, where each module has a particular functional role. For example, the Cas and DRG proteins here, they are scaffolding proteins that organize membrane-associated protein complexes. In these multi-domain proteins, each domain has a specific role in mediating interactions with other adapters and receptors. The Cas, MPP, and DLG proteins that we just saw are all members of a large called the MAGUX, shown here. And you can see that the domain content of these protein architectures represent variations on a theme. The proteins are similar in that they all contain a GLBIO domain and either an SH3 or a PDZ domain, and in most cases they contain both. And they differ with respect to domain order and copy number, and some of these architectures contain additional domains as well. This modular organization of multi-domain proteins supports rapid evolutionary exploration of new protein functions. So far, we've seen that multi-domain proteins are built from modular domains, and different combinations enable diverse biological roles. But the way domains are combined is clearly not the result of random sampling. Only a tiny fraction of possible domain combinations are observed in nature. And this suggests that domain co-occurrence and adjacency are stringently constrained. And many forces could be contributing to those constraints. For example, structural compatibility, mutational processes, and cellular environments. And in this work, we want to ask, how does protein function contribute to the constraints acting on domain architectures? We're going to use vector semantics to probe this question. I'm going to tell you how we construct domain and domain architecture embeddings, building on the analogy between natural language and domain architecture. I'm going to introduce 3 domain architecture techniques that use different information from the domain organizations we see. In vector semantics, words can be represented as vectors in a high-dimensional space, such that words with proximal vectors have similar meanings. And this is based on the distributional hypothesis, which poses that a word's meaning can be inferred from the words it typically appears with. So word embeddings are constructed by capturing the typical contexts in which each word occurs. And words that appear in similar contexts will end up with similar vector representations. Once we have vectors for words, we can also construct document embeddings. A common approach is to average the vectors of the words in the document. And this gives us a representation that reflects the overall meaning or topic of the document. And semantic similarity is now reflected by spatial relationships. Documents with similar subjects will have proximal vectors in the embedding space. Like natural language, domain architectures are also strings of tokens, and adapting the same logic, we hypothesize that domains that occur in similar contexts have similar functional roles. And so we construct domain embeddings by capturing their typical context in a set of domain architectures. We will see shortly how context is defined in each embedding we use and what organizational patterns they capture. And then we build architecture embeddings by combining domain embeddings. And here the hope is that functional similarity between domain architectures is reflected by the distances in the embedding. Domain architectures with proximal vectors tend to have similar roles. And this is just a visualization of the idea. Domains and domain architectures are represented as vectors. And in fact, this representation idea is not new. In the area of function prediction, some methods build domain embeddings, often combined with other kinds of information to predict protein function. And generally, better prediction accuracy is obtained when you combine other information. But those studies are interested in a different question than I am. Their goal is to obtain the most accurate protein function prediction. And my goal is to obtain a better understanding of the relationship between domain organization and protein function. And what are the implications for rules governing multi-domain protein design. Therefore, the way I train domain embeddings is to use only the domain architectures and not using amino acid sequence, not using labels. I want to capture whatever functional signal that's latent just in the domain organization alone. And now I'm going to introduce 3 domain embeddings we use. They define context differently, and this way they capture different kinds of interplay between domains. The first embedding is term frequency inverse document frequency, or TF-IDF. And here each dimension corresponds to an observed architecture. And the jth dimension of the embedding for domain DI is calculated as its copy number in the jth domain architecture divided by the total number of architectures that contain DI. And this way we capture how selectively DI appears across all domain architectures and emphasizing rare domains or architecture-specific domains. In short, the context here is domain architectures that contain DI, and domains that tend to co-occur in the same architectures will be close in the embedding. The second embedding is point-wise mutual information, or PMI, and here each dimension corresponds to another domain family in the vocabulary. It captures how much more often domain DI co-occurs with every other domain than expected by chance. And context is therefore adjacent domains, and domains with similar sets of neighboring domains will be close in the embedding. The third embedding is word-to-vec. It's a neural embedding model. For TF-IDF and PMI, the dimensionality is determined by the data. But in word-to-vec, the dimensionality is user-defined. We experiment with 3 different vector sizes, 5, 10, and 100. Unlike PMI, word-to-vec emphasizes co-occurrence patterns, but it does so using a shallow neural network trained to predict domains that appear near each other in a domain architecture. And in word-to-vec, the context of domain DI is the windows of the domains flanking DI, and domains with similar flanking domains will be close in the embedding. The embeddings I introduced all define context differently, but they all assume that context carries functional signal. And domain architecture embeddings based on these domain embeddings allow us to quantify functional similarity between domain architectures. And as a control, we introduce a null hypothesis where each domain is treated equally, and the interplay between domains is ignored. Under this model, functional similarity of 2 architectures is dependent only on their domain content overlap. We use the Jacquard similarity as a measure of shared domains between architectures. It's calculated by the size of their domain content intersection divided by the size of their union. Now that we have vector embeddings for domain architectures, we need a way to assess whether these embeddings capture biological function. I ask, do neighboring domain architectures in the embedding have similar functions? And I consider 2 scenarios where the embeddings do and do not share any overlap in domain content with the target architecture. And let's first look at cases where proximal domain architectures share at least one domain. Here we use gene ontology as our operational definition for function. And for any target domain architecture A, we find its k-nearest neighbors in the domain architecture embedding. We keep neighboring architectures that share at least one domain with A. We take the union of all Go terms associated with those neighbors as a prediction, and the Go terms associated with A is true label. And we can then compute precision, recall, and MCC, which stands for Matthews correlation coefficient. And for the empirical assessment, we use human domain annotations from superfamily. We have 5031 unique domain architectures in this dataset. The domain vocabulary has 1109 unique domains, and 4244 out of 5031 architectures are mapped to at least one Go code. And now let's look at how vector semantics perform in identifying functionally similar proteins. And here I'm showing you results for molecular function. I have 3 different values for K, and each row is one embedding, and here is Jacquard. And we measure precision recall and MCC, and there are only two things I want you to get out of this table. The embeddings consistently have better precision over the card, and here for MF, the card has better recall. And now if I show you the same thing with BP, it's the same story. Embeddings have better precision, the card gives better recall, except for k equals 1. And then for CC, embeddings still consistently have better precision, and the card has good recall. Why is it the case that embeddings give you better precision and Jacquard better recall? Our hypothesis is that Jacquard-based neighborhoods are functionally diverse. To assess this, we compare the total number of distinct Go codes in the neighborhood, in the shared domain neighborhood, obtained with Jacquard and with the various embeddings. I look at all three subontologies, and here I'm showing you k equals 3 and 5. And here for each row I have the embeddings and the Jacquard baseline. And what we find is consistent with our hypothesis. The number of distinct Go terms retrieved by Jacquard comparison is substantially larger than every embedding in all three subontologies by as much as 50%. And as a conclusion of this part, when we restrict our analysis to domain architectures that share at least one domain, we see that embeddings consistently identify more precise neighborhoods with fewer distinct Go codes, but more consistent in function than Jacquard. Does the same domain or the same set of domains always imply the same function? Here we see that it often doesn't. Using domain content alone generates broad hypotheses about the function. Embeddings refine this by capturing contextual co-occurrence patterns. And this leads us to ask the next question. For proximal domain architectures that share no domains, do they have similar functions? First of all, does this scenario actually occur? The answer is yes. Here I'm showing you for k equals 1, what fraction of such nearest neighbors determined by embeddings actually share no domains with the target architecture. And indeed, up to 30% of proximal multidomain architectures have no domains in common. And as expected, TF-IDF, which explicitly models domains shared across architectures, picks up fewest nearest neighbors among all embeddings that share no domains with a target. And so now we ask, are non-sharing neighbors functionally meaningful? To test this, here we have 3 columns for each embedding. We looked at the goal similarity distribution of these neighbors, and we noticed that many of them have very high goal similarity. Our embeddings identifying more such pairs than expected by chance. We compare the observed number of pairs identified by embeddings such that they share no domains and have a goal similarity score higher than 0.8 against the number of such pairs if the other architecture is selected at random. And across most embeddings, we have significantly more observed pairs with high similarity than expected. This is especially true for BP and CC. And for MF, the signal is not statistically significant, but the general trend holds. In most cases, we have more observed pairs with very high similarity than expected. And these statistical tests show that the embeddings are able to group functionally similar or related protein architectures, even in the absence of any shared domains. And these relationships are not random. How do they do this? There are 2 possibilities. One is embeddings capture functionally equivalent domains or domain synonyms, and the other is embeddings capture functionally equivalent domain combinations. To probe these two possibilities, let's take a closer look at 2 case studies. In our first case study, the Word2Vec embedding identified HOXP4 and TRPS1 as nearest neighbors, despite the fact that these domain architectures share no domain at all. They're both transcription factors, but more importantly, both of them are key regulators of the skeletal system development. And these 2 proteins both contain DNA binding domains, but different ones. Zinc finger for TRPS1 and homeodomain for HOXP4. We also checked sequence similarity directly. The highest scoring aligned region in a pairwise sequence comparison of these 2 proteins is just 5 amino acids long, and so there's no evidence of distant homology to explain this pairing. And the second pair are both involved in DNA repair and chromatin remodeling for nucleotide-dependent enzymatic mechanisms, and they both contain a few DNA binding domains and domains with enzymatic activities. Again, this pair has no significant sequence similarity at default parameter settings. So what we found is not due to missing annotation of a shared domain or sequence homology. And in summary, in both cases, embedding recognized architectures with shared functional roles despite having no domain in common. So to conclude this talk, the goal is to understand what part does protein function play in constraining multi-domain design. We experimented with domain and domain architecture embeddings that emphasize different contextual patterns. We found that embeddings capture functional relationships beyond presence and absence of shared domains. And these findings suggest the existence of functionally analogous domains or functionally analogous domain combinations, or both. So I want to thank everyone for their attention, and I want to thank the Drone Lab and my co-authors and my thesis committee and other people that gave useful suggestions on this project. And I am graduating soon, and I'm actively seeking postdoctoral positions in computational biology. So I'm happy to connect. Thank you.
Do we have any questions?
Hi. Yeah, that was really interesting. I was wondering, so is the intuition that because those domains can kind of appear in similar contexts in other proteins, that's why they're neighboring in this space, because even though they share no direct domain overlap, is that what's happening?
Yes, I think that's what's happening. So here, function is defined to be the context. So it's what other domains it appears with. Yeah, and so these two domains, they are not the same domains, and they have actually no homology, but they tend to appear with a similar set of neighbors. And so they are neighbors in the embedding space.
Yeah. Is it possible to kind of extract those, like, to kind of like give a rationale for why those two neighbors, like, is it possible to like pull that out automatically, the sort of like the kind of interpretation of why two things are near one another in space based on the model that you have?
Yeah, so so the question is, how do you say interpret why these two things are placed together in the embedding, and can you pull that information out automatically? Yes, so by capturing, so for the two domains that are close in the embedding space, you can extract the typical neighbors, and so there are different ways you can quantify, say, for example, by PMI, how unexpectedly they appear as neighbors. Yes, and you identify these sets of shared neighbors for these two domains. And then that way you can derive some intuition.
Cool, thank you.
Thank you.
Is there time?
Yeah.
Okay, thank you. Thank you, that was a great talk. So you said that TF-IDF works better with molecular function, and embeddings work better with biological process, sort of roughly? Is that it, or?
Yes, so for TF-IDF, it does have higher precision and MCC for molecular function when K is small. And so here I'm only showing you up to K equals 5, and as we reach further, so as you increase K to say 10, then the performance of TF-IDF drops significantly faster than other embeddings. So I think TF-IDF explicitly models shared domain content, and that's in some sense similar to Jacquard, what they capture. And so I think for a small neighborhood, for the one or two architectures that share most domains with this architecture, maybe the prediction potential is high for the function. But as you share fewer, well, I guess the drop in performance is significant when you consider domain content alone, or as that as your major information source.
So could it be that simply TF-IDF captures homology information better, and then which kind of works better for molecular function, whereas biological process is somewhat more complex to look at similarity, right? That's another problem.
I think that is definitely possible that TF-IDF is capturing that more.
Very interesting. Thank you.
Thank you.
Thanks.
Okay, let's give